Good morning students, myself Dr. Shweta Jain. Today we are talking about extracellular matrix. Learning objectives. After this lecture you should know the definition, what is extracellular matrix, functions, components of extracellular matrix, abnormalities in diseased state of extracellular matrix. Now, what is extracellular matrix? As the name suggests, extra, that is means outer the cell is extracellular matrix. So, it is a web of protein and carbohydrates at cell surfaces. Cells and organs surrounded by a glue-like substance. In figure, you can see the extracellular matrix, like collagen fibers are there, elastin fibers are there, glycoprotein and proteoglycans are also there. Now the next we will look at the functions of extracellular matrix. So first is structural integrity, cell matrix interactions, proteolytic turnover, cellular responses and growth factor regulation, which is through sequestration and activation. So mainly it provides support and encourage for cells. It regulates and determines cells' dynamic behavior, that is polarity of cells, cell differentiation, adhesion, migration. It provides mechanical support for tissues and organ architecture, that is for regeneration and healing process, determines the and maintains the structure for growth, and it is also place for active exchange of different metabolites, ions, water. Now we see what are the constituents of extracellular matrix. It consists of structural proteins, specialized proteins, proteoglycans. So in structural proteins there are three types: collagen, elastin and fibrillin. In specialized proteins this fibronectin and laminin. Proteoglycans can be syntecin, aggrecan and versican. Later on, now we discuss all the structural proteins in detail. So first we will start with collagen. So collagen is most abundant protein in animals. It is secreted mostly by connective tissue cells and in small quantity by other cells. Collagen contributes to the stability of tissues and organs. It has a great tensile strength. It maintains structural integrity. So, collagen is the main components of tendons, bone, fascia, ligament and skin. Plays an important role in tissue and organ development, in cell differentiation, polarity and movement. Now look at the collagen structure. There are about 28 types of collagen which are known, and they are made up of 30 types of polypeptide chains, each coded by a separate gene. So collagen is a repeat unit of glycine XY unit. You can see in this figure. So it is a chain structure. Glycine XY repeats in a left-handed turn. So here X is proline, Y is hydroxylysine or hydroxyproline generally, and these both proline and Y, that is hydroxyproline, they makes the chain more rigid. And glycine, because of its small nature, is able to be accommodated in the crowded helix. Tropocollagen is the structural unit of all collagen. Each tropocollagen consists of three polypeptide chains called alpha chain. Tropocollagen has a quaternary structure called triple helix where three alpha chains are arranged into right-handed triple helix and each individual alpha chain have a left-handed helical structure with three amino acid per turn. So it is a rod-like structure which is about 14 nm in width and 300 nm long. How the collagen is synthesized. So collagen synthesis can be of in two steps. That is, first there will be intracellular synthesis, then the second is extracellular synthesis, and thus the collagen synthesis is completed. In this picture we can see the intracellular synthesis. So first is the synthesis of pro-alpha chain. Here pre-pro-collagen on ribosomes occur. You can see pre-pro-collagen is forming on ribosome. Second is the hydroxylation of selected prolines and lysines. So here the hydroxylation is taking place in second step by lysyl 5-hydroxylase or prolyl 5-hydroxylase. Here the hydroxyl group is attached. Third is glycosylation of selected hydroxyl lysine. Glycosylation of selected hydroxyl lysine is there. So after hydroxylation, there the galactose or glucose unit will be attached there. Addition will occur with the help of galactosyl transferase or glucosyl transferase. So you can see here the glucose unit. So glycosylation occurs. Now the next step, that is the fourth step, is the self-assembly of three pro-alpha chains. Now these three pro-alpha chains will get self-assembled and pro-collagen triple helix formation occurs with the formation of disulfide bonds between the registration peptides at both ends of the pro-collagen. So you can see here the disulfide bonds are formed. After this pro-collagen triple helix formation, the next step is secretion of the pro-collagen. Now how the secretion will occur? It will be by exocytosis into the extracellular space through the secretory vesicle. Through the secretory vesicle, the exocytosis will occur and these are secreted in the extracellular space. So from first to sixth step, that is till fifth step, and then the secretion occurs. These all processes are intracellular, mainly which are occurring in endoplasmic reticulum or Golgi compartment, and then the secretion occurs through secretory vesicle. Now after secretion, the extracellular synthesis will occur. What will happen? This pro-collagen molecule is there which will get cleaved. So cleavage of propeptides occur. In cleavage of propeptide, it is catalyzed by the pro-collagen peptidase. So pro-collagen peptidase will cleave it and a tropocollagen is formed. After the formation of tropocollagen, there will be oxidation and deamination with the removal of NH2 group and formation of covalent cross-links catalyzed by lysine oxidase. Afterwards, the last step, that is the eighth step, will be the self-assembly into fibril. And self-assembly will occur After that, aggregation of collagen fibrils to form a collagen fiber will occur and collagen fiber is formed. So this is all about the collagen synthesis. These all are the steps which I explained you earlier. Now we will look the types of collagen. So there are basically three types: fibril forming, fibril associated and network forming. So in fibril forming collagen there is type 1, type 2, type 3, type 5 and type 11. Type 1 and type 5, they are present in bone, skin, tendons, ligaments, and type 2 and type 11, they are present in cartilage, intervertebral discs, eyes. Type 5 is as same as type 1 as I explained, and type 3 is present in skin, blood vessels. Fibril associated. So there will be two types in it, type 9 and type 12, and the third one is network forming, which will have type 4 and type 7. Now we will see the collagen disorders, which are called collagenopathies. So mainly there are three types of collagen disorders: Ehlers-Danlos syndrome, then osteogenesis imperfecta and scurvy. These all are due to any deficient defect in the synthesis of collagen. Now this is a diagram where you can see at what step which defect occur and leads to the cause of collagenopathy. So in Ehlers-Danlos, there is deficiency of collagen processing enzyme, and thus the Ehlers-Danlos occur. In scurvy, it is due to there is lack of hydroxylation as hydroxylation is inhibited, and in osteogenesis imperfecta is after the glycosylation the step is defective due to the deficiency of enzyme. Now Ehlers-Danlos syndrome. So it results from a deficiency of collagen processing enzymes, for example lysyl hydroxylase or pro-collagen peptidase, or from mutations in the amino acid sequences of collagen types 1, 3 or 5. In the picture you can see the clinical features. This is stretchy skin, hypermobile joints, vascular defects can be there, fragile skin can be there. So this is all about Ehlers-Danlos syndrome. Now the next defect is osteogenesis imperfecta. This disease, known as brittle bone syndrome, is a group of inherited disorders distinguished by bones that easily bend and fracture. There are around 8 types of osteogenesis imperfecta, but type 1 is the most common. Here you can see there is scoliosis curves. Now come to scurvy. It is due to deficiency of vitamin C, which is also required for synthesis of collagen. Scurvy often presents itself initially as symptoms of malaise and lethargy, followed by formation of spots on the skin, spongy gums, and bleeding from the mucous membranes. So, in ascorbic acid deficiency, interchain hydrogen bond formation is impaired, as is formation of a stable triple helix. And collagen fibrils cannot be cross-linked, greatly decreasing the tensile strength of the assembled fiber. Patients with scurvy also often show bruises on the limbs as a result of subcutaneous extravasation of blood due to capillary fragility. Here you can see in the pictures the spongy gums. the patches on the skin. So that's all for today's session. Then the next leftover extracellular matrix we will cover in the next session. Assignment for today's session is, what is extracellular matrix? Write its functions and components. Second one is biochemical synthesis of collagen. And third, describe diseases associated with abnormal collagen synthesis. And these assignments you have to complete in your notebooks. Thank you.